Today I'm going to be talking about the system that I'm developing, which you guys have heard a good bit about, but also mostly diving pretty deep into why we're doing the stuff we're doing and what an actual full stack looks like to support neural experimentation, and in particular, to try and get some of the handicaps off. I'll go over a little bit of the thought behind it. If anything doesn't make sense, just ask me to explain it. It's probably my fault. So yeah, so the basic question that's really guiding all this is, say you have a neural culture, right? It's a neural network, a real one, sure, but it's just a neural network. And the real question is, how much control do you have over it, right? Because this entire project we're actually trying to compute with them, or maybe you're a medical researcher, right? And you're trying to look into different properties of your neurons, right? But whatever it is that you're actually interested in looking at, the key insight is every bit of information is contained within that network, right? And eventually it gets made external if it's in a live animal, for example. The neurons will actually be connected to muscle tissue so that the activity of the network causes external events. If it's in your MEA, right, it might trigger some of your other electrodes to do something. But the real insight is that every bit of your computation is completely contained within your network state, right? And so the real challenge as I see it, is two things. It's one, how well can you extract information out of your network? And two, how well can you influence the information in that network? And right now we're at a bit of a poverty with both of these because these things are hard to work with, right? They're really, really small. And it's not like a simulation, which is actually where we want to be. Friedhoff knows a good bit about that, but in the simulation, not only do you have access to every relevant variable that you want, you can also reach in and change it at any time, right? And so in a really, really abstract way of thinking, you can think of this neural network not as just a static thing, but as a series of different distinct snapshots that's changing over time, right? And so what you've got is at any point in time, you have an entire network full of information and state. And so what we actually care about when I'm saying how much control do you have over it, the ideal situation would be akin to a simulation, right? Which is at all times, you've got access to all network information, right? So you can see the membrane potentials, you can see the synaptic weights, any relevant parameter you can think of, you can extract, right? Obviously, we're not going to get this. But the reason why I'm setting this up is to show you this is where we're eventually trying to hit. And then we're going to work backwards to what we actually have available and what we can do to get closer. And then finally, also, we really want to have control to alter any part of this network at all times, right? Because again, we're thinking of this neural network as some kind of a computation that we can influence from the outside, and we can look at and inspect based off of whatever information we're able to extract. Our degree to which we can manipulate this object is largely gated by how well can you actually manipulate the information that's inherent in that network. And so going to something that's a little bit more of a realistic model, right? We're going to have our neural culture, and through these electrodes, that information is going to get brought out to our computing stack. And right now I've just black boxed it, right? And I'm just trying to say, okay, let's think, what does this computing stack need to accomplish if we want it to be as general as possible? And by as general as possible, I really mean to give us as much control as possible over this culture. So we can get further away from something that looks like a real culture where we're hamstrung and we're trying to get at it through indirect methods, and more looking like something like a simulation where you have an idea and you can actually just go ahead straight away and implement it, right? And I've just recopied the ideas from the last slide, which is your ideal is you want to access all of your network information at all times, and you also want to be able to alter any part of the network at all times, right? We're obviously not going to get that, but let's see how close we can get. And so in a realistic setting, what we're actually interested in asking ourselves is how much information can I extract from my network, and how much control do I have to alter my network? And these two questions are really what's going to guide the entire systems development and why we're actually doing the stuff we're doing. So starting off with this first question here, how much information can I extract from my network? Well, first, let's look at how are we actually getting information out? What's our best idea right now? And where are we losing information, and what can we do about it? So, you guys have probably seen this a good bit of times, but I'll go over it real quick. At least our basic theory and the assumptions that we make when we're working with these MEAs is that, we know neurons communicate with electrical spikes. You put two neurons next to each other, they're going to fire electrical signals between each other. And if you put an electrode in between these neurons, you can actually read out the voltage on that electrode, and you can detect these spikes, right? And so the implicit assumption here is we've already done a huge collapse of information. We're saying we don't care about the synaptic weights. We don't care about the shape of the cells. We don't care about exactly how the synapses are propagating. We're just worried about what neurons there are and when they're sending spikes. And this actually isn't an unreasonable thing to do, in particular because we don't have much choice, right? Take something like a synaptic weight. How would you measure a synaptic weight? Right? There's no clear way to do that for an entire network at once. And so what we're going to do is we're going to work with the best thing that we have. And so to extract information right now, we're just putting as many electrodes as we can in a tight area and trying to measure all the spikes that are firing around in our network and trying to infer something out of that. And then the final point here is just that obviously, we want more information about our network. So the more electrodes we can pack in here, the better in terms of extracting information from the network to do something useful with. Now comes the hard reality, which is the actual information loss, which is we lose, even just focusing on spikes and neurons, we lose a ton of information in these networks right now. In terms of coverage, so real quick, just imagine you have just one electrode for a second instead of multiple in an array, right? And you've got a bunch of neurons around it. And the question is, well, how many neurons is that electrode actually picking up? Or if you want to think in an MEA setting, think out of the total neurons in your culture, right? What percentage of them are actually being read out by any one of your electrodes? And it actually depends on your culture. It depends on the cells you're using and a couple other factors here. But just an estimate from Jim Smith, using a neocortex in particular, was you only get about 2.5% of neurons, right? So you're already losing 97.5% of the available information after we've already sliced it down to just spikes, right? So we have a fraction of the information, right? And I just put this here just for something to think about, because these things I'm not going to cover with this work, but just for anyone who's interested or would like to think more about it, something like a CMOS MEA is what this is actually trying to resolve, right? Which is they're trying to stop bleeding out so much information by packing as many of these electrodes together as they can, right? So they're not missing neurons and they're not missing spikes. They still do, but they miss it less than we do. The other problem is aliasing, right? Which is this electrode here, if it, say it picks up all 5 of these surrounding neurons spiking, how does it actually know which one's spiking? And the answer is it doesn't, right? It picks up somewhere in the neighborhood of 10 to 100 neurons. And you're already losing information there, right? Because if you consider, again, your network, the inherent information we're saying all we care about is the neurons and which spikes they're sending. And in particular, we only actually care about 2.5% of them because that's all we can get. Well, we're further reducing that now by saying, but for every pocket, every electrode pocket, we kind of have to treat it as one neuron because we don't really know which one's firing and which one isn't. There's a possible solution to this, right? You guys have all heard of spike sorting, which is trying to analyze the shape of the spike and then see, okay, well, which neuron do I think actually fired this? And you can do some k-means clustering and try and get back some of your information that you lost here. But either way, you're probably not getting a ton back. Another problem is, the further this neuron is away from this electrode, the weaker the spike is. And so at a certain point, you don't actually know if you're looking at noise or at a spike. And so there's some pretty hard problems to solve on the edge here. And finally, the coverage and aliasing does actually scale with electrode size. This is quite intuitive if you think about it. If your electrodes get smaller and you pack more of them in together, you're covering more of your culture, right? Aliasing will also get better if your electrical neighborhood shrinks as you're shrinking these electrodes. However, for this work, electrode size is actually pretty hard to scale. And we're working with traditional MEAs. We're not working with CMOS MEAs, right? And I'm working on the computing stack side of this. So I'm just taking the electrode size I have as a constraint on the system. So the electrode size is what it is. There's certain things you can do to get back information, but we're not going to focus on them. I just want you guys to be aware of, where you're actually losing information when you're trying to record from these cultures. Well, so the next logical question then is, okay, so we've got this really, really reduced set of information, right? How good of a system do we have to build to actually get all that information out, right? Because we want to figure out how good does it need to be, set our target, and then build a system to fulfill those requirements. And the 2 things I'll talk about first is what resolution do you need when you're recording, and what resolution do you need for your stimulation? Stimulation is a little different, so I'll get to that second. For recording, right? What we're really asking is, say you have a spike somewhere on your line, right? How fast do you have to record samples to actually detect that spike? This is more of just an experimentally determined number, but 30 kilosamples a second is, for all intents and purposes, is going to catch you all the spikes you're possibly going to catch, right? Put another way, this means if your system can record at 30,000 times a second, it's not going to do you much good to record it 60,000 times a second, right? Stimulation is a little different. Stimulation is, and you guys have all probably heard of rate-based versus time-based hypothesis for how neurons are actually computing. The important thing is that neurons can actually detect pretty precisely when the spike arrived at them, right? And it actually depends a lot on which neuron you're talking about. But they're generally, most neurons are precise to about 100 microseconds. Some neurons, auditory neurons will get lower because they need to locate exact distances. But for most neurons in your brain, you're looking at 100 microseconds and up, which, you convert this to a rate and you factor in Nyquist and you get about 20,000 times a second, right? So if you have 20,000 different windows in a second that you could send in stimulation if you wanted to, you've effectively covered all windows for the neuron. To the neuron, it doesn't make any difference if you're able to send it in at 40,000 different steps instead of 20,000, right? So put another way, if you can record and stimulate at 30,000 times a second, you've basically extracted all the relevant information that there is to get here, which is good news because Inten's chips actually already do this for us. So we don't need to worry about this constraint in particular. The one that we do need to worry about is the last real constraint, which is where there's actually a pretty good bit of discrepancy right now, which is, okay, we've talked about, how do you get information out of your network? What resolution do you need for sending information back in? Well, the question is, okay, well now how do you generate that information you're sending back in, right? How do you actually generate that stimulation and what are the constraints on that? And in particular, I'll skip this first bullet point, that should have been later. We'll go to delay right now. In particular, what we're asking is, okay, say you record something in your culture, right? And you want to be able to respond to it. How fast do you need to be able to do that before it doesn't really matter anymore? At which point, you're not actually gaining anything in terms of your experimental setup. This is actually kind of an open question, but the best estimate is you can either use the width of a spike or the propagation delay between two neurons. Depending on which neurons you get, you can get lower numbers, you can get higher numbers, but roughly you're looking at somewhere in 1/10 of a millisecond to a couple milliseconds, right? And so put really, really roughly, if you're around 1 millisecond turnaround time, you've basically extracted all of the usefulness out of your experimental setup that you can, right? And so that's going to be the target number then, is you want to be able to take in a recording, process it, and decide what you want to do for your simulation within 1 millisecond. And the good news here is that modern CPUs run much, much faster than this. So we actually have a lot of wiggle room here to implement this if we do it well enough, right? And then finally, the one other constraint is that this computing stack needs to permit general deterministic computation, right? Because we're trying to build a capable experimental device. It's not enough to have some fixed function logic here and say, okay, you want to implement something else, go build another machine, right? So that covers general. Why does it have to be deterministic, right? There's a lot of different things in a computer that can actually, it can impact whether or not you get done on time, particularly when you're talking about milliseconds and fractions of a millisecond. The problem with that being, if you write an algorithm that naturally assumes the hardware is going to do what you tell it to do, right? And it violates your assumptions, well, what do you do with the rest of your experiment? Say you're running a 2-hour experiment, right? It's not immediately clear how you recover from that. And so it is pretty important that this computation is deterministic, at least to a fairly reasonable probabilistic bound. Turning all of this into actual constraints looks something like this, where we've got some goals and then we've got some features to address this. The first one we went over, which was, what do we have to do in order to not lose any more information? We need to be able to record and stimulate at 30,000 times a second. And like I said, luckily, Inten's commercial head stages can already do this. We're using Inten's head stages. They're pretty good. That covers our first problem. The second part, right, is we need this to be general and deterministic for how you're going to process your recorded data into an actual stimulation command. The general part basically means you need a CPU somewhere in the back end. The deterministic part means you need a real-time CPU, or at least one that's capable of real-time execution. And right now, that means we're using an RPU for our back end. We just talked about, having to have a 1 millisecond turnaround time to get from your recorded data. Yep. What's that? Just real-time processing unit. It's a CPU with real-time features. So it's got a couple of kilobytes of 256 kilobytes of onboard memory, where you can choose to only interact with that memory. And because there's no caching, you actually know when your memory requests are going to return. It's got a couple other things like that. Basically, you can get a hard guarantee that if I compile the C program, I actually know how long it's going to take. And it's not that, after a certain number of tries, maybe I get an outlier that messes up my experiment. The reason why that's important is because you're probably going to be running, say you're running at a 1 millisecond loop, you're running that loop 1,000 times a second. And so if you do an experiment over a couple hours, even a small probability of failure is still too much. And so you do need some determinism there. This is probably the most important constraint, which is, again, once you get down to about 1 millisecond turnaround time, you're probably not getting much more out of going too much further lower. How you actually get to that is pretty hard, but at least what I've put in for this slide to address it is probably the most important part, which is hardware accelerating the spike detection task. And if I go back a couple of slides here, we've got two neurons that are communicating together, and every now and then they'll fire a spike. Yeah. Yeah.
But the microprocessor, whatever you're using, it's to make the computer work that fast, it's not a problem. But you have to have these delay lines, the connections.
Yeah.
And those can ruin your whole day because they can be slow.
Mm-hmm.
So I'm not sure if you're, putting the constraints on the CPU, it's not going to be a big deal. But the rest of it, the recording, for example, writing it to memory or whatever other storage device you can find, that has to be timed so that it catches, be fast enough for it. So
yeah, I'll get to, in just a slide or two, I'll get to actually all parts of the system and where the data is actually moving, and that'll probably make it a little clearer. But you're right, the CPU itself is less of a concern. The way I look at it more is, you're right, it's the other parts of the system that are actually going to dominate your latency. And because this is a latency sensitive operation, that basically means the CPU gets what's left. So it's optimize as much as you can on the rest of the system, and then see what you can give to your users, basically. The first most important thing though is, the CPU, you're right, you can clock it pretty fast.
to record fast enough
Yeah.
to catch whatever spikes or whatever.
Yeah. 30 kilosamples a second. And the really hard, so 30,000 times a second is not that fast to a modern processor, right? If you're running at 4 gigahertz, running in 30 kilohertz is not actually that fast. But it is that fast if you want to scale to thousands of channels, which we do. So you do actually need to be pretty efficient then. And most importantly, say you're doing a couple thousand channels, and you're recording. If you're using Intel system, you need to be you're recording 32 bits for every command that you send it. Really quickly, you get into gigabytes a second territory. And the important insight is that most of that data is totally useless, right? Most of that data is just the neurons sitting around and doing nothing. And that's kind of the whole point, right? That's why they're energy efficient. But from a computation perspective, right? From this computing stack's perspective, it doesn't actually care about seeing raw data, unless you have a very unusual program that says, hey, I actually want to do something weird with these raw voltages. Most of the time, this computing stack just wants you to pass when the spikes occurred and on what channels they occurred on, right? And the insight here is that's a really, really regular task that parallelizes very well across channels. So it's a really good candidate for hardware acceleration, basically. The reason why that's really important is, if you're compressing your data, and spike detection will get you somewhere from 10, it'll get you a couple of orders of magnitude, one to two orders of magnitude compression. And what that means is, in the limit, you're going to buy yourself 1 to 2 orders of magnitude more channels, provided you have enough hardware to actually support it. So the takeaway there is your spike detection has to be in hardware.
So Jakob, I have a question for you. So how would the system recognize which is useful information, which is not? Which spikes are meaningful versus the spikes that the neuron are just doing, like end-plate potential, just firing for no good reason?
So right now, there's no good way to discriminate that, actually. So the way that I'm having my system look at it currently is all spikes are relevant to the processor.
Mm-hmm.
You are right, though. There's probably a good bit of intelligence that could go into that, into saying not all of these are probably very relevant to what we're doing. Whether or not you can put that in hardware, I'm not actually sure, because that might be task specific. But yeah, right, there's compression to be had there that we're not taking advantage of right now.
And one thought could be that, is it that when many of those electrodes are showing synchronous bursting versus just random firing, so maybe when they are cooperating, at least locally cooperating, that might have some more meaningful. So I'm thinking in terms of critical, subcritical, supercritical state of the network. So if they're near critical state, then there may be a bunch of neurons in specific locations which might be filtered out, right?
Yeah, no, you're totally right. The hard part about this is, deciding which spikes are important and which ones aren't is pretty task specific. It depends on what you're actually doing with your network. And the whole point of this work is to try and get away from having to do task specific stuff in hardware, because that's really slow and hard to do. We want to do it in software because you can turn it around very fast and have a nice closed refactor loop. The problem is, like Lauren said, the time is actually dominated by the non-CPU parts of the system. And so by the time you've put the spikes into the CPU, it's already too late. You've already burnt the time getting them. And so it's, you would have to figure out some way to discriminate in hardware if you actually wanted to save the time.
But if you want to discriminate in hardware, that's great, but it's not going to lower your requirements for the hardware, because you still have to read it. Process some way to say, no, I don't like it.
Yeah, it'll save you on the data movement into the CPU and back out again, but that's it. You're right.
Yeah, but that's
and either way, it's I should have put better numbers in here, but I'll tell you guys numbers throughout the presentation. Most of the time it is dominated by your filter. The reason being, any kind of a digital filter is essentially trying to do some kind of a smoothing effect where it wants to look at a window and suppress certain frequencies. Unfortunately, say you have a really low frequency, well, you have a really long wave, and you have to look at a pretty wide area to actually determine whether or not you're seeing that frequency. And so there's actually a time trade-off in the filter that's just completely inherent. And so to a certain degree, if you want good spike detection, you're almost always going to have to wait somewhere in the realm of half a millisecond to a millisecond just on the filter. You can get that down quite a bit if you're willing to cheat on your filter a little. So there's some wiggle room there. The very last thing I had was, and we already went over this. This thing really needs a scale well channel count, right? Because that's kind of the goal, is we want to put a whole lot of channels on this culture. We're already losing a lot of information. We need to get back as much of it as we can. So we need this thing to scale as well as possible with channel count. So when we figure out how to put more electrodes on this thing, we're not hamstrung, right? Which means, and this is also, fairly obvious from the rest of this, but the channel data path really does have to be in hardware. You can't throw all this through a CPU or you're going to have problems. This is an abstract sense of the system, and I'll show you what it actually looks like in a second, but just an abstract overview of the system. You've got your neural culture, and you've got electrodes ferrying it into your computing stack. So from your computing stack's point of view, right, you've just got electrodes coming in that can record and electrodes going out which can stimulate. And in our case, since we're using Inten's RHS system, these electrodes are actually aliased. Each electrode is capable of recording, and it's capable of stimulation. In your chain for your computing stack, we've got Inten's commercial head stages here, although theoretically they could be any head stage that's capable of recording and of stimulating. We've got our FPGA and we've got our RPU. Real quick, just going over what these things are actually doing and why they're there, right? So the head stages, you actually need something to convert from these raw analog voltages into the digital domain. Inten has commercial head stages, they're miniaturized, they're already pretty good, and they're capable of 30,000 times a second on recording and stimulation, right? So they already take care of those first two constraints for us. So that's really handy. The other thing is they also handle all stimulation. So we don't need to worry about on our computing stack, hey, how are we actually generating the right waveforms and what kind of a current source are we using? We can kind of forget about the analog picture and just think purely about digital samples coming in and stimulation commands going out. So we're kind of abstracting up the ladder here to where the computing stack really gets cut off here, where it's the FPGA and the RPU together. And the FPGA is really just a way of saying hardware accelerating common tasks, right? And in particular, what it's doing, is it's taking in these digital recorded samples, right? And it's processing it into just spikes for the RPU to see. So its almost its number one job is just pass spikes to this thing. We don't actually want it to see anything else. Obviously for experiments, and confidence in your data in general, you should save the raw samples. I haven't put it in here, but this FPGA will also pass the raw data out to some external storage, right? So you'll still have access to the raw data afterwards, but you don't want to have it within your loop. That's going to slow you down. Yeah.
You said something very interesting before, or maybe some, I don't know. Yeah, no, but there is something that I still delayed in my brain. You said that these neurons, most of the time, they do nothing.
Yeah.
Do you account for the fact that they do nothing? I mean, it looks like like sparse zeros, in a sparse storage, zeros, they do nothing.
Yeah.
So can I get rid of them? Can I compress it away?
Yeah.
I have also some idea on maybe what.
You mean can you press away can you compress away the samples or?
No, they said that they do nothing. So when they do nothing, why record them? Why deal with them?
Yeah, so this is actually pretty similar in spirit to the previous question, which is, is there a way to compress this even further by only looking at the important parts of your recording? The hard part is how do you actually determine when it's doing nothing? The conservative approach is just record the whole thing, and you're right, you're probably recording a lot of stuff you don't need still. The hard part is how do you actually determine which of you could have some kind of a timer on each electrode.
But you mean you also record noise? You have noise?
Yeah, well, there's always a little bit of noise in your recording, right? There's always going to be a little bit of noise. But if you're not, if you have an electrode sitting there and none of your neurons around it are firing, you're just seeing the whatever noise line you have come out. And hopefully what that should translate to is the head stage will actually have to record all of that, and it will pass all of that off to your FPGA. But your FPGA will then run spike detection on it and just throw it all out. So you're still taking in a lot of data you don't need, right? The hard part is how do you determine which data you actually don't need,
But some of them is not just white noise or whatever,
Yeah.
right? So that is harder to figure out.
Yeah.
But if it's just noise, you could filter it out.
Oh yeah. Well, spike detection is essentially just filtering. Spike detection is a fancy way to say run a filter, run an absolute value or an emphasis algorithm, and then threshold it. It's essentially filter and threshold. It is really all spike detection is doing here.
That's on all channels.
Yeah, that's on all channels. So it's
quite a bit of operation.
no, you guys are right. There's a lot to be gained here. Right now I'm doing, I guess, for lack of a better word, the simple and dumb but efficient thing, which is just record all the channels all the time. Yeah, well, it's efficient in terms of not in terms of in the system, no, but in terms of me making it so you guys can use it, yeah, it's efficient. Because otherwise I'd have to figure out, how do I actually determine which is useful and which is not, So it's right now I've chosen generality over potential speed, but you're right, there is a good bit of opportunity there where there's probably some processing you could do in the loop where you take a look at your system, and if you notice we haven't seen a spike on this thing, or say you have some analysis algorithm that's trying to estimate how much are these spikes actually contributing to the network activity, and if you can tell it's not doing anything, you can probably turn those off. This also gets into a good bit though of it's tough because it's where the network grows, right? Where we don't have a ton of control over exactly where that network grows. So you won't know this ahead of time. This has to be something that's done while you're experimenting, if that makes sense. So yeah, that covers the FPGA and then the RPU. All this thing is really doing then is it's taking these incoming spikes from the FPGA for granted, right? And it needs to somehow process this into outgoing stimulation. Again, this has to be general and it has to be deterministic. And what that actually looks like right now is the RPU will allow a user-written C program to take in these spikes and produce some kind of a stimulation. Now, obviously, there's some constraints, right? You can't write an infinite while loop in this because then we can't actually guarantee that it's going to finish in time. Oh yeah, I can't see it on my screen. Sorry.
Do you want to ask your question? You should be able to unmute.
Your hand is up.
Okay, hi. Thanks a lot. And sorry if my question doesn't make sense. I'm coming from a very, I guess, human EEG focused background. So one beauty of how networks compute in biological tissue is that the information is not actually in the spikes.
Yeah.
I mean, it's a topic that's still debated, but the way, well, biological networks are really good at integrating information at a given level and then moving it on to the next level. So it's not like if we only knew every little bit about every spike of every neuron in the brain, we would know what the neuron is doing. And the reason we can't do that is that biological networks have emergent properties that are different from the collective knowledge of the individual spikes. So what neurons do is integrate all that information. And now again, what could complement this kind of spiking level readout would be focusing more on the fields that reflect the collective properties of the tissue. I mean, there's a lot of internal debate within the neurosciences about whether ephaptic coupling is an informative mechanism beyond spiking, but even separate from ephaptic coupling, meaning coupling outside of the synapses between neurons. Even beyond that, the larger question is, do the local field potentials carry more computational information than individual spikes do? So I think this very spike-focused problem, which is there's way too much information, might on the long term be better solved by biology itself. And we would intersect with it at a higher dot level than at the individual spike level. Not sure if that makes sense.
No, that makes total sense. I should have mentioned, sorry, I'm not covering it because it's less relevant for this, but I do plan to have LFP available on the RPU. It's a much slower reading, right? So it's much less data than the spikes. So for the actual performance critical parts, LFP just isn't really in the critical path. So I haven't focused on that, but you will have LFP available as well. I do actually agree with you to a good bit that I don't think that you can just say, we'll just round up all the spikes and everything that's going on, right? Like from the start where I said, okay, you have no idea what the synaptic weights are, right? You don't actually know how the neuron interpreted the information it got. The reason why I'm focusing on it so much, is it might not be perfect, but it is something that we can get really good information extraction on, right? Where we can at least extract large amounts of information from the culture, and assuming we do it fast enough in a good enough system, right? We'll at least get a lot out. Where it's not clear to me how you would look at LFP and determine, okay, well then how did the neural system actually compute that result, right? Where if you're looking at it from a really granular level, how do you actually infer what went into the process of doing that, if that makes sense?
Right. I think that sounds great in terms of being able to understand the mechanisms of how the neural population decided their output. But at the end of the day, the larger goal of the overall project, right, is to not have very temporally high resolution, very spatially high resolution data, because that gets us back to needing silicon computers, right? The whole point is to replace them and have a simple output where the computation of this vast amount of spatial temporally detailed content is actually done by the biological tissue itself. But I totally agree that in the intermediate time where you want to understand mechanisms, this totally makes sense and is really amazing. I just wanted to say that, keeping that larger goal in mind and making sure that LFPs are not just yet another thing that we look at, it might be one of the primary signals we need to read and focus on and understand as the emergent property of the tissue. Just wanted to emphasize that out there too, but I totally agree with you.
Oh yeah, you're completely right, actually. I'm, don't tell NSF this, but I'm actually much less worried about exactly how you make the biological computation run efficiently, and I'm much more interested in how is it actually doing the computation, and just the general underlying feeling I have is it's really not clear to me at all how I'm going to make a biological system compute if I don't understand how it's computing, and so this is just like you said, it's much more geared to a tool into actually trying to figure out what's going on than it is into actually trying to do some energy efficient computing, if that makes sense.
Absolutely. And you're probably going to be able to answer questions that in biology directly, like in vivo, could never be answered. So this is really exciting in terms of the mechanistic question you're posing.
Yeah, you're right. Eventually, if you want to run something energy efficient, you probably don't want to monitor every single neuron in the network. That's way overkill. And hopefully, eventually, we do get there. That'd be nice. Yeah. Yeah. So this is what it actually looks like. This is actually an old picture, but it's just the best one I had. Going back one slide, this is actually reversed. So this neural culture is over here on the right in the MEA. The head stages are over here. You guys have probably already seen them, but for those of you who haven't, they're actually plugged straight into the MEA. And the FPGA and the RPU are both together on this chip, which is the zinc ultra scale. The rest of this stuff is all just a whole bunch of machinery to get signals in and out of the board. We're actually not using this little green piece here and that little blue piece here. This is just an interface to try and connect to these Intel RHS chips. And unfortunately, there's actually no standardized way or no good way to get signals from those RHS chips into an FPGA, which was actually really frustrating. Intel actually sold a part to do it, but it didn't work. So instead, we made our own. This is made by Chase, so I don't think he's here because he's working on a paper submission. But he's really good at what he does. And he put together this first draft of it. We're probably going to go through another revision of it. But essentially, it's replacing this green piece here and this blue piece here. And it's essentially directly connecting the FPGA out to these RHS head stages. We're also probably eventually going to look into making a better version of this. And potentially down the line, the head stages should also be modified. I'm not going to talk about that a ton today because we do actually have a hard out in 15 minutes here. But there's a whole lot of room for improvement in this system. What I'm making here is just the first attempt to get something down so you can build something on top of it. And then finally, I'll talk just a little bit about what's actually going on inside the thing. A lot of stuff, big diagram, a lot of stuff on the screen at the same time. But essentially, the basic model of what's going on here, and just to draw it back to reality, we've got our head stages down there, and most of this entire diagram is taking place on this chip right here. This chip right here is the actual green head stage converter, right? These are actually different now. We don't use these two parts because they didn't work. So these two parts we've replaced with the custom piece we made. But the general idea is the same, right? This system is interfacing to the cultures through the head stage. And then it needs to also actually hook up to a host PC for control and to transmit data back so you can actually read it out. I won't go too into detail with it, but the one thing that I actually wanted to highlight with this is the system should be somewhat resilient to changing different parts of it, right? And so in particular, these intent head stages, most of this code that's actually running in the FPGA doesn't depend on the head stage. There is some head stage dependent code up here because you actually have to understand what stimulation you're capable of, right? But the head stage, we shouldn't be tied to Intent's head stages, right? So it's not completely independent of head stage, but a large bulk of the code doesn't actually care about what head stage you're using. In addition to this spike detection has been isolated. The reason for this, and this is really what I wanted to put this slide in to talk about, is unfortunately, we have no idea what spike detection algorithms are good and what are bad. We can empirically run them and see what seems to perform better or not for tasks. Say you want to run, you drop some way to do an MNIST task on your MEA, right? You can run different spike detection algorithms and see which ones give you a better answer, right? So that's an indirect method of trying to look at it. But there's actually no good ground truth spike detection data set out there. And the reason being, it's really hard to get ground truth recordings of neurons in bulk. So our options right now basically are we can do extracellular recording of a lot of neurons at once, which again, you have the problem of is it noise or is it a spike? Or we can record just one neuron and we can actually know exactly what it's doing, right? And unfortunately, when you put those together, you actually don't get ground truth. And so the end result of this is I've intentionally cut the spike detection algorithm out of everything else. And what I'm putting in right now is my best guess, but it's still a guess. The other thing that I thought I'd say just on the compression side is, right now Intent's head stages are reading out 16-bit voltage readings, right? If you actually think about the sparsity of the data that's coming out of the network, right? Really, it's just it's a very simple event. It's every now and then you get a spike, right? So the actual data content over, say if you have over every second, you have one spike, right? You need enough information to store the time of the spike in the channel, but you don't need 16 bits 30,000 times, right? So there's, in terms of how you actually detect this efficiently, there's almost certainly much, much better ways to go about this. I think the thing that's actually blocking us right now is because we don't have ground truth. We don't know how aggressive we can get with this, right? But my general intuition behind it is assuming you have an experimental setup with low enough noise, you can probably get very, very aggressive with your spike detection because it happens infrequently enough that you're gathering a whole lot of data that you probably don't need. I think that's about all I have for today. Just the last thing I'll do is a real quick update. Right now where we're at is the system's capable of recording. So it's interfaced properly with Inten's head stages. You can record through it. We're not actually sure, there's still some noise going on it. So we got to figure out, how to denoise the system a little bit. But once we've denoised it, it should be pretty much fully capable of running recordings. One thing to note is we are still running on a CPU right now, these APUs up there. We're still running it through a CPU because that's much easier to debug on than an RPU. So where we go from here is, we finish up getting the recording solid and making sure we're actually getting good data out of it. And then we can move into actually putting these full pieces in. I think that's about everything I got for you guys. So any questions?
Jakob, what does FPGA mean? Those who are not from ECE.
Oh, sorry, field programmable gate array. If you're not in ECE or computer science, the way you can kind of think about it is it's just reconfigurable hardware. So normally when you make hardware, it costs a ton of money and you're stuck with whatever you get, which means you have to do a whole lot of effort up front to make sure your design is really right. But in FPGA, you can just flash it each time you make a mistake. So it's a pretty cheap and inexpensive way to prototype out hardware acceleration.
Hey, Jacob, can you hear me? Yeah, hey, thanks for that presentation. It was very accurate, I think, in so many things, and almost distressing for me to hear because it makes me think we're not going to be able to really listen to these guys or control any of their stuff. And I think if you develop a system that can respond at the millisecond time scale, roughly the time scale of a spike, then that will be certainly something better than other people in the world have right now. I mean, that would be a fabulous device to have. And there's all kinds of ways you could probe the system with that. That said, I have a couple things that might be somewhat hopeful. So one is that it turns out that if you, and many of you may already know this stuff, but I just want to throw it out there for discussion. Many of you may know that if you have a brain-machine interface where someone's recording from human neurons, you can get by decently well controlling the robot arm with only like 100 neurons. So it might not be that you have to record everything. And the second thing about it that's somewhat related to this is that even if you take a really good neural network simulation, you can destroy a decent number of the neurons and the thing will still behave relatively well up until some point, so they're robust to lots of damage. So it might be that really what's going on is there's just a ton of redundancy in the system, and we might be able to get away with things if we just sample a decent number. So that's point one. It's just the number issue of the neurons. The second point I want to make is just the time. So it is true that the spikes are operating on the millisecond time scale, but many animals have behaviors that operate on timescales that are on the order of seconds or even minutes. And so it might be that we might still be able to get away with understanding how things work, even without having to go down to the spike level timescale. Maybe for local computations you need that, but for anything that the animal's doing, a brain is basically tasked with outputting some sort of behavior that's maybe on the scale of seconds. So those two things give me hope that maybe we can get there. Just some comments for discussion.
John? Yeah, go ahead.
I'm done.
You're right on the first one there. The coverage, 2.5% is a really, really scary number when you first see it. But you're totally right. Really what it does is it just puts us on the same playing field as the neurons. It's not exactly similar, but you can kind of think of these electrodes in the MEA as just the neurons we actually control, right? And the fact that you see a lot of behavior in the brain where you can have, some input signal that is not a lot of neurons, right? And it'll propagate through the network and grow and do something, involving a large population that you didn't actually need to, you didn't need to stimulate the entire population to do that. You just need to stimulate the small part of the retina to do it, right? So this actually isn't, I don't think it's much of a handicap once we actually know how to use it. I think what it's a handicap for is for actually trying to figure out how it's working. And that's a big thing to say, but the basic idea behind it, is we thought, let's just figure out how one neuron works. And once we know how one neuron works really well, we'll just infer how the network is going to work, and we've solved everything, right? And I forget who was saying it earlier, but someone was talking about emergent properties. What we found out is that's actually not what happens at all. Unfortunately, you can know one neuron's dynamics pretty well, and you still don't really understand what happens when you put a lot of them together. And so the hope here is, or maybe less hope, but just the intuition is, okay, well, let's go one level up, right? And let's start looking at networks of neurons and try and understand really well what makes them tick and how they work. And will that give us everything? Honestly, probably not. The fact that there's emergent properties, like you said, over several seconds means there's probably more going on here than recording a few thousand neurons in an MEA and inferring out thought and consciousness. That's probably not going to happen. But you will get a lot more insight than you currently have. Can't remember the second point. Sorry, can you remind me real quick? I had some.
Yeah, it was just behavioral timescales are not millisecond.
Yeah. Well, I guess, I got both of them, I guess there. Yeah, I guess the response I have is in the end, we're probably not after something that looks like this. This is kind of an in-between thing because we want to know how it's working, if that makes sense.
Right.
Think of it more as a microscope than a CPU, if that makes sense.
Yeah, that's a good response. I like that. Yeah, thank you.
Lawrence, you got one? I think we got time for one more here.
One more question. Did
Yeah.
you think about testing this thing? I mean, making maybe some contraption or something like this that validates what you're recording?
Yeah. So Xantel actually had a really good idea for this. Because we don't know whether or not it's actually right. Right now, we're still just denoising the thing.
Yeah,
Yesterday we
a
had
lot of noise. You have to see what if you
Yeah.
record it as noise or
no,
wishful noise or
we actually
whatever.
have that right now. So yesterday we had a bunch of line noise, and Xiaotian actually helped me figure that out. So we got the line noise out of the way, and the noise actually looks pretty much what like Intan's controller picks up. But how do you know? And so we actually, me and Xantel ran a couple of recordings today. And we got results that look like spikes. The amplitude is about half the size it should be, but the width is right, and the shape looks generally like it does on the Inten thing, but how do we actually know? So we got to do some more work in trying to figure out where the noise is coming from, or if there even is noise. But the real gold standard test that was his idea, which is a really good idea, is have an MEA where the electrodes split out, right? So one electrode, 2 wires, and plug our device in and then plug Inten's device in and record at the same time. And make sure you don't stimulate, but as long as you're just recording both at the same time, then we can actually go back and time match them up afterwards and get a pretty good guarantee that at least we're doing what Inten does. And as long as we do what Inten does, that's already industry standard, we're fine.
So you can come up actually with a bunch of these if you want sanity checks.
Yeah.
Make sure that.
Yeah, the other option, would be like have a saline solution and have some external power source to hold it at a constant voltage relative to your ground, and then measure and make sure you get the right one. I think it's probably better to go with this one though, just because, well, it's easier because we already have the device. And then it also tracks changes, so we can also see not just can it record one number right, but is it actually temporally accurate as well? So yeah, there's a lot of hardware difficulties that go into this. It's not as pretty of a picture as really nice looking diagrams. But we're getting through them. We've gotten past the interface problem, and we're getting through the noise right now. So I don't think we're actually very far off of just being able to record whatever we want. And from there, software moves faster than hardware. You're pretty familiar with that.
Yeah, I think the points that John and Sepide raised, I think there is a bigger question for me. I think what you're doing is fantastic to look into like a sub-millisecond time frame temporal readings and maybe even stimulation if possible. But you know, there is an issue about emergent properties where when many entities work together and they go to a different state, and then the different state, which in this case may be a synchronous activity of, let's say, 10 neurons, maybe 100 neurons, like John was referring to. So individual spikes may be not relevant even information. Again, I'm just speculating this one, rather than how these 100 neurons cooperate and behave synchronously, giving rise to these second or 10-second time scale events. You know, a classic example of this emergent property is water becoming ice. So when water is going through 0 degrees temperature, then all the molecules are interacting with one another and becoming ice. But if you want to understand the behavior of ice, we don't need to go and understand the molecules of water before they became the ice. So once the system goes to a new state, in this case, many neurons firing in some cooperative way, that information is actually what is more relevant than what individual neurons were doing. So when we talk about ice, we talk about its hardness, elasticity, you know, conductivity, and so on. Nothing to do with water molecules anymore. In fact, we may not be able to understand the properties of ice by looking at individual water molecules anymore. And there's a famous article, less is more, or less is more. So I think, myself included in my lab, we've been focusing a lot on looking at individual spikes from each electrode. And each electrode is reading the spikes not only from a single neuron firing, but also, you know, the action potential going by, and who knows what it is really reading. It's reading a local field potential, right? Locally. It's coming from many neurons, single neurons, it's action potentials going by. I'm just thinking maybe we should think slightly bigger scale and seeing what other information can you collect with much less energy, yet makes more sense out of it. I'm just putting a point in regards to what John and CPD was talking about.
I think one of the big problems at play here is that there's not much translation between scales, unfortunately, where and there's so many, there's a huge breadth of scales in the brain. Not just scales, but also diversity. But the scale problem, I think, is the big one here, where, you know, when you single neuron to small network of neurons is totally different, and small network of neurons to enough to get emergent properties is also totally different. And so, you know, it's which ones do you study them at? I don't think it's totally water ice.
No, it's like a trivial example, right?
No, I do agree with you that I don't think we're going to get everything out of this, you know. But it is easy to measure, at least with the existing technology. You know,
right.
I actually do agree that, you know, like I said, the worst leap was not losing 97.5% of our spikes. You know, the worst leap, I don't have a great graph. Oh, well, the worst leap was going to the MEA model, where we've already given up on all other information that's inherent in that network, right? But at the same time, you know, this is because this is where the technology is at today. You know, that this is you know, there's the other option is you know, develop a stack that works on emergent properties, which is really fuzzy. So at least from a computer guy's perspective, you know, it's much easier to work into the existing stack. But I do think you guys are really right on this, that, you know, you're not going to solve everything by looking at every single spike. What you might do is get a little bit of an insight into how our neurons actually, you know, how are they actually building small scale networks together? And from there, you might get a little bit of insight into, okay, now how do you move into coordinating those smaller scale networks?
Sure, yeah.
But
okay.
But I understand that you're looking at each one of them separately, and ideally you would get some model which you can use to put them together and get the higher level, you know, of abstract whatever,
Mhm.
you know, simulation or whatever you want, but higher level.
Yeah.
And we may be looking for that for a very long time. You may be, you're saying is I need to understand that. And obviously,
Yeah.
if you would have all this information and make all these hierarchical models, you would understand how it all works.
Yeah,
But we may not live long enough for that.
no,
So
maybe a medieval captain.
why I think that he's laughing too much about that. But at any rate, we won't. So we just agree that we are not going to understand everything.
Yeah,
And well, I'm going to make a joke that the machine learning guys do not need to understand everything. In fact, they maybe don't understand anything, and that still it works.
yeah. I mean, we're-
Doesn't
you know,
bother them.
Yeah, you're. I think we are probably further out than most people think on this stuff, unfortunately. The good news about this, though, is assuming we don't move away from a channel and voltage sample model. This thing is meant to be general and that it can go down to those timescales. If you want to work at a higher timescale, you totally can. Right. The problem I'm trying to address right now is that you literally can't go to the smaller scales. And so I'm trying to make that available. But there's nothing stopping you from working at a higher scale and averaging out.
But when you measure, for example, the various potentials or whatever it is, right? You're measuring them now individually. And then if you decide that you want to go at a higher scale, you're not going to measure them individually. You're going to measure maybe a cumulative effect directly.
Yeah, yeah.
And so it says you ignore all that stuff. It says, okay, there are 1 million electrons, right? But you just measure the voltage on the capacitor, the one that they charge.
Yeah.
And you leave it alone.
Yeah, I mean, there's also other ways to integrate that pretty cleanly as long as you're willing to vary what your definition of a channel is, you know. Also, you know, it might pain you to hear, but you can totally get that information by integrating this information. You're doing a lot of useless work, you know, but if the system is capable, why do you really care about burning a couple more watts of power, you know?
I think that what you're doing is perfectly fine right now. And we'll see if we can use that,
Yeah.
or if we move on to something, you know, more general or whatever.
Yeah, this is.
I wouldn't listen to all these people who tell you don't do that.
Well, this is going to be a very, very simple model at first. You know, like the idea is build a simple base system that we can understand and then build on top of it. You know, so for example, the very first thing we're probably going to build on top of this is adding support for optical stimulation. You know, so letting you run a straight signal out of the FPGA to trigger your optical stimulation whenever you want. You know, so there's a lot more to be added, but hopefully it's useful. We'll see. Any more? All good? Awesome. Well, thanks for listening, guys.